Okay, great. So yesterday, I think we used this. After termination, we started looking at post-transcriptional modification and a statement that newly synthesized RNA molecules are not immediately functional and they may require modification. So today, after this summary on what we are expected to see, we're going to look at the details, especially on how 5' capping occurs in mRNA, modifications that are very important in tRNA, as well as the splicing of rRNA in both prokaryotes and eukaryotes. So talking about modifications in eukaryotic mRNA, the 5' capping, we are basically going to see how there is a 7-methylated guanine M7G cap is added to the 5' end of the transcript, and how to identify the poly A tail, how the adenylation occurs, and why it is important. Then also looking at how introns and exons are separated in a process known as splicing. So how best can you define splicing? What is splicing? What are exons? And what are introns? So all these are topics, like detailed topics that we are going to cover. Then of paramount importance is the maturation of tRNA, knowing that we have 20 amino acids that can be arranged in different ways to form a specific protein. And yet we only have, and then we have like 64 codes on the genetic code language, of which 3 are instructions: the start, which is AUG, and then the stop codons. So that leaves us with, we have like 60 of them remaining as a message to carry a particular amino acid. So how do the transfer RNAs actually identify their specific amino acids? So the structure itself, the cloverleaf structure, which is similar in all the tRNAs, and then we have the acceptor arm, which has a specific modification of CCA, then anticodon arm, which is different for the tRNAs. So the anticodon arm, which is the arm which will have a message, a triplet base message opposite to that of the mRNA, and so they can best pair to read which amino acid that tRNA should carry. We'll discuss this much as we enter into translation itself. We'll need to start with that before we discuss really how translation occurs. And all these other different modifications. RNA is important as components of the, whether in the small subunit or large subunit, and these ribosomes are different in prokaryotes as well as in eukaryotes. And last time in class I mentioned what the S stands for. The S is not additive, so it's how heavy or how. So when this RNA has been produced, eukaryotic mRNA requires processing. So we are going to see the 5' capping, 3' polyadenylation and splicing before it can become functional. And apart from it functioning, it means it has to also be transported from the nucleus to the cytoplasm. The prokaryotic mRNA is mature immediately, so it doesn't have much modification. But tRNA and RNA also require modifications. So let's now look at the actual processes. Yes, someone raised a hand. Please just speak up because I can't really see you.
What is meant by capping? 5' capping.
Yes, so that's what we are going to see now. What is 5' capping? So this is where we put a 7-methyl guanine group or a cap on the 5' end for reasons of exporting the RNA from the nucleus as well as protecting it from the enzymes. that can degrade it, known as exonucleases. So now let's look at the post-transcription modification. So this is actually a very long topic, but I'll just discuss much of what I think we need. The other stuff we can read as out of interest to really enjoy it. So let me just show us this. I hope we can watch this. So this is transcription in prokaryotes.
The central dogma of molecular biology: DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time.
So what is coming out there is in yellow is the RNA. So behind it, we can see the DNA reanneals, so it doesn't open completely.
Through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay. So what about in eukaryotes?
In eukaryotes, RNA polymerase II transcribes protein coding genes. A basal level of transcription requires a set of basal transcription factors that bind to DNA or other proteins. First, a protein recognizes and binds to the TATA sequence at the promoter. TATA binding protein, TBP, bends the DNA. Auxiliary factors bind to the TATA binding protein. This protein complex promotes binding of RNA polymerase and its associated proteins. The complete pre-initiation complex denatures the nearby DNA helix. RNA polymerase II and its associated proteins move along the unwound DNA and produce a messenger RNA complementary to the DNA template. The level of transcription is increased above the low basal levels by activators bound to specific enhancer sequences. Activators can act to stabilize the
Okay. So let's now look at the post-transcriptional processing of RNA. So we want to see how RNA is actually made to function at the end. How does it function? So the initial transcripts that are made, when you hear the word primary transcript, you're simply looking at what has been transcribed, what has been synthesized directly from DNA. So the term transcript in molecular biology simply means RNA. So the primary transcript in itself is the initial molecule of RNA produced. Sometimes it's known as heterogeneous nuclear RNA, or abbreviated as hnRNA. So it is heterogeneous because it still has other things attached on it. It's not the final RNA that we are interested in. So there are things that should be removed from it, spliced out. So it's still heterogeneous in nature and is still in the nucleus. So most of the modifications for that are found in the eukaryotic one transcription compared to prokaryotic. So in prokaryotes, we have DNA, RNA, protein. in the cytoplasm, they occur together. So as DNA is being transcribed into RNA, protein can also be translated from RNA within the cytoplasm. And this is very fast because there isn't much protection of the messenger RNA in the cytoplasm. But in eukaryotes, it happens in the nucleus. So in eukaryotes, this is referred to as nuclear RNA. And it has to pass through two processes, which are modifications and transportation to reach the cytoplasmic RNA. So the RNA that goes to the cytoplasm is mature, it's protected, it's covered. So we have different types of processings. So like I mentioned, these slides are quite detailed, but I'll make emphasis on what I think is best for us to know. But the interactive will have a lot of information for you to do and help you understand. So when we look at the types, we have three main types of RNA processing. There is cutting and trimming to generate the ends. So this happens in all the three main RNAs that are directly linked to translation, which is rRNA, tRNA, and mRNA. rRNA stands for ribosomal RNA, a component of the ribosomes, which are the site for protein what? Synthesis. Then the tRNA, transfer RNA, the RNA responsible for transferring amino acids to the ribosome so that we can have peptide bonds that will allow the linkage or synthesis of polypeptides, which are proteins. Then we have mRNA, which is the messenger RNA. This is the intermediate between DNA message or the gene message from DNA and the protein that is being synthesized. So this is the middleman. The messenger RNA carries the information from DNA to protein, since DNA doesn't leave the nucleus and DNA has to be maintained intact, so it shouldn't be interfered with, apart from carrying or copying this information to RNA, which later on can be degraded once the protein has been synthesized. The other type of modification is covalent modification. So this is where we have addition of a cap on the 5' end and a poly A tail on the 3' end or hydroxyl end of the messenger RNA. So we add the methyl group to 2' hydroxyl group of ribose in mRNA, rRNA. Then there's extensive changes of bases in tRNA, like a number of bases are changed. And this gives different tRNAs their capabilities on how they function. We even have tRNA that is responsible for carrying the first amino acid when it comes to translation. And depending on the organism, it can be methylated or unmethylated. So all these changes of bases in tRNA is very important. And lastly, we have splicing. Splicing occurs in all the three, pre-RNA, pre-mRNA, and pre-tRNA by different mechanisms. They are not going into details about the mechanisms because I think it's beyond our scope in this course, but I'll just make mention of them. You don't need to memorize them, but just understand how the basics on how it happens. So in prokaryotes, the splicing occurs for pre-RNA and tRNA, and not because the mRNA, the genes are produced without or limited introns, so they don't need splicing. But for eukaryotes, because the genes that are being expressed may be separated by longer sequences of intervening bases, they need splicing so that they are brought closer together and be allowed to be transcribed. So let's start with transfer RNA synthesis and processing. It involves quite a lot because we have all these base modification as well as folding. Always remember that RNA is single-stranded, whether it's tRNA, mRNA, or rRNA, but they can sometimes be folded, and that creates different structures in them. So starting with the tRNA, we have different tRNA molecules that are encoded by different genes, and this depends on what the tRNA is going to be transferring, which amino acid is going to be transferring. So in prokaryotes, the mature tRNA molecules are generated by processing longer pre-tRNA transcripts. and this is through a specific step involving RNases. RNases are enzymes that can actually degrade or cut RNA. And they are in prokaryotes, they are called D, E, F, and P. So you don't need to remember these D, E, F, and P, but know that they are exo and endonucleases. Exo means they cut on the external ends, either the 5' or the 3' end of the RNA. And endo cut inside, like along the length of the RNA. So these can cut exo or endo, external or inside. And because they cut nucleic acids, they are known as nucleases. And the nucleic acid they cut is RNA. So generally, they can be called RNases. So remember, they are enzymes that are able to splice or cut the RNA at specific site. So after transcription, the nascent or the pre-tRNA has to fold to form a structure known as stem and loop. And the general name for that structure is what? Clover leaf. So since it has to form this stem and loop structure, it's very straightforward in prokaryotes. It's much easier in prokaryotes. Someone's mic is on. Maybe ask them to mute because they're giving me feedback. Yeah.
You know, I don't know about you, Paul, but 4F5s seem very personal to me.
Yeah. Okay. So what about steps in eukaryotic tRNA maturation? Remember, in eukaryotes, it's a bit more complex. And so even the factors involved in the maturation of tRNA in eukaryotes are complex. So talking about tRNA molecules in eukaryotes, they are synthesized with a 16 nucleotide 5' leader. So this 16 nucleotide 5' leader is simply bases, nucleotides, 16 of them that will be on the 5' end before we even reach the actual nucleotides that will be involved in the structure of this mature tRNA. So these are used in order to allow the further modifications that will need to be done. Then we have the 14 nucleotide intron.
Hey!
Please, whose mic? Is it?
Madam,
Is
just
it?
remove
Who
them
is
from the
it?
group.
McLaren is the problem. Last time it disturbed man.
Okay, he's muted now. All right. So for the eukaryotes, they have the tRNA molecules are synthesized with a 16 nucleotide 5' leader, meaning these nucleotides are found on the 5' end. And they're very important when it comes to modification as they identify they'll identify the arm of this tRNA. And then we have a 14 nucleotide intron and extra 5' and 3' nucleotides. So this whole primary transcript has to be converted into a secondary structure that is going to have stems and loops, the D arm, the anticodon arm, all those loops. So it has to be folded in such a way that it forms that clover leaf shape and identifies which arm is going to be transferring the amino acids or the amino acid attachment site. So when we compare it with that of the prokaryotic tRNA, the 3' terminal end and the 5' end of this eukaryotic tRNA, they form this arm which is known as the acceptor arm. And we find the modification of what? A CCA that is added. The base CCA is added on all the tRNAs and ends with a hydroxyl group. So this is added by a special enzyme known as tRNA nucleotide transferase. Please, you need to know the name of this enzyme. At least know that we have tRNA nucleotide transferase is It helps us to understand that it identifies that this is the site that will be able to transfer the what? The amino acid. Nucleotide, meaning when we look at transcription, we'll see how the tRNA has to be activated. This is where the amino acid that is going to transfer is attached to the specific tRNA. So there is need for this enzyme to actually add this group, the acceptor arm. So the name of the enzyme is tRNA nucleotide transferase. Know this enzyme. Then introns, like the first bullet states, there are a 14 nucleotide intron in there and some extra nucleotides. So the introns are removed by endocatalytic cleavage. What does it mean? It means that those enzymes, the nucleases, RNases, are able to cut inside and remove or cleave. So sometimes this cutting, we use the term cleaving. So it removes them, it cuts them off and like gets the parts that need to be joined to mature tRNA are linked together. So the enzymes are able to do this. So there's endocatalytic cleavage followed by ligation of the half molecules, meaning the intron part is removed. So this is how the nascent can look like. So this is how the pre-tRNA looks like. It has all these loops, all these foldings, and so we have the 5' end where it shows, but then our 5' end doesn't need to have an extra loop. So the dark pink color here, this should be the structure of the mature tRNA. The pale pinks there were just part of the transcription because they were needed. Some of these parts could be identified as where the promoter region is, and then the gene for this final tRNA that should be there was somewhere inside along the DNA sequence. And since the promoter is further away from where this gene is, it means any sequence between the actual gene and the promoter region are also transcribed, but they are not needed in the mature tRNA. So that's why they have to be removed. So for example, from the 5' end, we see endonuclease. So RNSP can cleave off that part to leave a 5' end that is needed. Towards the 3' end, again, we see where the endonuclease can actually cut. And then we have the exon nuclease, such as RNSD, can actually cut that part. Then we have the enzyme, the transfer peptide, that transferase, coming in to bring or add the sequence, CCA, that is needed on the 3' end. Then we have all these other base modifications. So for instance, we have the methylguanine, where the guanine is methylated in that arm. We have the P there representing pseudouridine, where we have a pseudo or a different image of the uridine. We have the 4-thiouridine, so represented by the T's. So all these modifications. IP, we have 2-isopentyl adenosine. So where we have adenosine, we have all these functional groups added or changing the structure of the base. These are all important to form a mature tRNA that is going to carry a specific amino acid. And remember, out of the 20 amino acids, we don't need all the 20 tRNAs. We just need half as much to allow the transfer. So let's just look at the steps, like I've already mentioned. You don't need all these, you don't need to memorize all these, that RNSF does this. You can read in your own time. But just to run through, we have factors, the RNases. So they're just named F, D, P, based on how they are discovered and what they do. So there is cutting of a flanking sequence at the 3' end, like we are showing here. So there is this cutting of the flanking end, and this generates a precursor with 9 extra nucleotides, which are then removed. 7 of them are removed to leave the 3' nucleotides where the the CCA is going to be what? Added. And then there's further trimming on the 3' end. So when we talk about removal of the splicing, like I mentioned, we have endocatalytic structure. The introns can be of different length depending on the pre-tRNA. So they can be of different length, but they're usually very short compared to those found in mRNA. So you're talking about 10 to 20 nucleotides, and they have no consensus sequences. So they are not necessary. They may be removed to allow what? To allow a proper tRNA maturation. And remember that all these processes of splicing or cleavage involves energy. And the energy that is used in this case is what? ATP. So there is ATP hydrolysis that occurs. Even in the ligation of the ends after splicing, there is ATP hydrolysis that is used. So this is very important. Remember the energy. And let me make emphasis about the CCA addition at the 3' end of tRNAs. So all tRNAs have this sequence, triplet sequence of CCA. So they all end with CCA. So amino acids are added to the CCA end during charging. So when we talk about translation, we'll learn or discuss about what charging of tRNAs is. Unless a tRNA is charged, it cannot be used in translation. It cannot carry amino acids to the ribosome. So the CCA is very important for charging of tRNAs for translation. And so most eukaryotic tRNAs, the CCA is added after transcription. So after removing all the parts that are not necessary for a mature tRNA, at the 3' end, hydroxyl end, the CCA is added. So this is in a reaction catalyzed by tRNA nucleotide transferase. So the addition of this CCA means this particular tRNA is ready to actually carry amino acids to the ribosome. Once the amino acid is attached or the tRNA is charged. So how does this happen? So this is a structure, schematic diagram of the tRNA cloverleaf structure. And this is known as secondary structure because of the foldings forming these different arms. So the cloverleaf structure itself, the CCA, we have amino acid binding site. And then we have the anticodon site. Here I have XYZ. This represents the anticodon that will be complementary to the codon on the mRNA in the ribosome. Then we have all these different arms, the D arm, the pseudo arm. These can differ from one tRNA to the other. But what is common is that all tRNAs, they carry the CCA arm on the 3' end, and they have an anticodon arm. So these are important. They are present in all what? Transfer RNases in the secondary structure, and their secondary structure is cloverleaf. tRNAs have a cloverleaf. This is very, very important for us to know. So I already mentioned about base modifications, and just for us to remember is that modifications in 20% of the bases, the modifications are that they actually account for unusual bases, 20% unusual bases, like the addition of the methyl groups to the base or sugar, converting like adenine to methylated adenine or methylated guanine. We also have where uracil is reduced to dehydrouridine. So it forms something different, or it can be converted to pseudouridine. So someone once asked me about this. This is means pseudouridine. So this is where uridine has been converted. It has been changed, and it's an enzyme that can actually convert this uridine to uridine. So the enzyme itself is known as pseudouridine synthase. Then there can also be deamination. This is where the amino group is removed from adenine. So when the amino group is removed, remember the structure of adenine. So removal of that amino group produces inosine, I, or hypoxanthine. So that changes the base in the tRNA. And these are quite important to the functioning of the tRNA and how it will carry the amino acids. So on my right, on my left, I have the normal bases that are found. And then on my right, we have modified bases. So all these different modifications. So for uridine, we can have all these 4 different types of modifications. Cytidine, we have all these different types of modifications. So I don't expect you to memorize these, but it's okay if you have the time or interest to just see these modifications, but at least remember that we have these modifications like inosine, the methylated guanine. These are quite important for us to see because they are very common. So this is the final structure of a mature tRNA. So you can spend time, try to draw it on your own and see if you can label these parts, the amino acid attachment site, the CCA, the 5' end has a phosphate there, the D loop, T loop, variable loop, modified purines, where can you find them, and even how to label the anti-codon loop. Know this cloverleaf structure, draw it, label it from your head. Yeah. So now let's quickly look at processing of mRNA in eukaryotes. So when you talk about processing of mRNA in eukaryotes, we should always remember that in prokaryotes, there is little or no processing of mRNA, and know the reason. Your exam or test are multiple choice, but it's expected that you know the stuff. You don't just guess. So know why there is little or no processing in prokaryotes, whilst we have intensive processing in eukaryotes. So processing of mRNA in eukaryotes, prokaryotic mRNA is degraded very fast because transcription and translation occur almost simultaneously. So because it's not protected, the cytoplasm contains a lot of enzymes, the RNases, that can actually degrade the mRNA. So it has to happen that when we have transcription, almost immediately, within a few bases of transcription, translation starts. So because of this, we expect that there shouldn't be much processing. The cell cannot wait to begin processing, otherwise the entire mRNA will be degraded. So the mechanism is that ribosomes are assembled on unfinished mRNA, a lot of them. And the first histone can be translated very fast. The first gene that needs to be formed into a protein. And so because we have many ribosomes assembling on the mRNA, they form a structure which looks like strings on a bead. So it's known as a polysome. Polysome meaning that multiple ribosomes on a string. So this string is actually the mRNA. And so we have the same protein or the same gene being expressed at the same time. So it's easy to make multiple copies of the protein before the mRNA is degraded. So the internal citrons are partially protected by the stem loop structure that is formed. So the mRNA in prokaryotes forms this stem loop structure in its preterm to at least protect the internal genes. So that's why we have flanking sequences of the transcribed or the nonsense mRNA, which are not even carrying what? Any gene that needs to be translated. So if those are exposed on the terminal ends, the main genes of interest, they are mostly inside or internally located. So that gives them some protection and allows the mRNA to be translated before it's finally completely transcribed, before even termination. But when it comes to eukaryotes, RNA polymerase II transcribes different genes from the nonsense RNA genes, and it forms the heterogeneous RNA which undergoes processing to form mature mRNAs. So you find that single genes have their own mRNA, whilst in prokaryotes, the single mRNA can have multiple genes on it. So they are cistronic. So they have many genes which can be referred to as cistrons on the same mRNA. So the mRNA in prokaryotes, please write that down. mRNA in prokaryotes is called polycistronic because it has many cistrons or genes on it that are transcribed together and translated together. In eukaryotes, the mRNA has single genes. So each gene can have its own mRNA. So they are what? Monocistronic, meaning it's one cistron at a time, one gene at a time. Although some genes can actually be transcribed together, but it's not common. In eukaryotes, what is common is that each gene has its own mRNA. So this is a very large picture. It has a lot of information which we may not really manage to understand now. So allow me to just say, you find time, you look at it after explaining some concepts so that it becomes easy for you to understand this. So here we are looking at mRNA capping, polyadenylation, and splicing. So talking about pre-mRNA. Pre-mRNA is covered by three important proteins that form what is known as hnRNP. hnRNP is an enzyme. This is the heterogeneous RNA protein. So it's a complex that is used to process the mRNA, to allow the capping, to allow the splicing, even identifying it, that it's actually what? It's nascent, it's new, it's immature. So now when we look at these proteins, you don't have to memorize them. The A, B, and C proteins, they form the hnRNP, of which this hnRNP can actually attach with the other proteins and it forms the particles that we now can identify as snRNA molecules. So these are denoted different names, or such as UI, U2, you don't have to know this. So all these complex together with more specific proteins, and they form this snRNP, which is again another complex used to identify how the mRNA is going to be spliced or to be cleaved. And in determining the methylation sites of pre-rRNA, so they are also involved in pre-rRNA, identifying where it should be methylated. So all these different complexes, different proteins and enzymes, they work together in the processes. Talking about 5' capping, soon after synthesis of about 25 nucleotides long of mRNA chain, the 5' end is modified, and this is a chemical process. So there is addition of a 7-methylguanosine, M7 gene, M7G rather. So it's important to know what this stands for. So this means okay, someone is busy messaging. So this means 7-methylguanosine. And this is added by the enzyme mRNA guanyl transferase. Again, if you are taking notes, write this down. The enzyme that is responsible to chemically modify the 5' end of mRNA, which is known as 5' capping, is mRNA guanyl transferase. So the mRNA guanyl transferase adds a 7-methylguanosine on the 5' end. Remember, the 5' end has a phosphate group on it. So it adds this 7-methylguanosine. Name of the enzyme, mRNA. Always remember it's mRNA because it's multiple choice. You may find that it's written as tRNA or rRNA. So remember it's messenger, mRNA guanyl transferase. This enzyme is responsible for the 5' capping. Then D, we have 3' cleavage and polyadenylation. So remember in that nonsense structure, we had the pale pink showing all that long structure on the 3' end. That part has to be removed and then polyadenylated, meaning addition of A's, adenines, a poly tail, a long tail of adenines. So the mature 3' end is generated by cleavage at polyadenylation. How does this happen? A specific sequence in the DNA as the mRNA is being transcribed, a specific sequence in the DNA and this pre-mRNA transcript. So we are seeing the 5' AAU, AA3'. It provides the adenylation signal. Do we have to know this? Yes. At least know the adenylation signal. So the adenylation signal is 5' AAUAA 3'. So this is in the DNA, meaning this provides the signal that this is where the pre-mRNA needs to have a long tail. Then we have about 250 nucleotides long poly A tail that is added by a poly A enzyme. So this enzyme is known as poly A polymerase, PAP. PAP is polyadenylation. Now, this is added to the nascent mRNA. So polyadenylation therefore marks that it has a 5' cap. It comes after the 5' capping. So once the 5' cap has been added, the 7-methylguanoside thing has been added, then the 3' end also has to be protected. So this is when the poly know the signal. So looking at the time, I don't know, we missed a lecture on Monday because it's a holiday. Can we suggest when we next enough time for when can we meet for a makeup to finish this topic? Sorry?
What about Saturday, madam?
9 to 10, I have the 4th years, then 12, I have the 3rd years. So maybe afternoon, what time can we meet?
14.
15. 15.
Which
15 hours.
14 hours.
No, 14 at quarter mathematics.
15 have math.
Can we meet at 16 then?
16.
Yes, no
16
problem.
hours.
Yes,
Because
please.
16 hours, I'll be home. So it's easier for me to have this. So tomorrow we can meet at 16 hours. Okay.
And
Yes, and
madam,
we are kindly asking for the notes, madam.
Are you
madam,
guys,
Manoj,
are you visiting Moodle? I was told that the outlines like this, they're not notes. Don't go around saying I gave you notes. We're not allowed to do that because this is a shared course. We have 8 lecturers. So use the notes on Moodle, but you can use my lecture outlines to help you as you study. Okay. Yeah.
Madam.
Okay, so I'll share these lecture outlines. Yeah. Okay, thank you very much. Have a good
You can subscribe, like, and share for more lectures. I promise I'll upload all the lectures on my channel. As long as you share, you like, yeah, I'll upload all the lectures. So do that as your part, and I'll do my part. Thank you.